So I just quickly wanted to share some tips from the lab perspective on successful lab testing. My disclaimer is that, whatever lab you work with, you should confirm these kind of things with them. But I think this is generally applicable no matter who you work with. So I wanted to go first through how to sample and then submitting and what you should expect. So how to sample, I mean, the key is getting a representative sample, and you've probably heard that before. So I just want to walk through a couple of scenarios. THC compliance, this is going to be something that is state dependent. So for Virginia, what you're going to want to do is take 3 to 5 inches from the top of the plant and combine. You want to take 4 random cuttings from a single strain, and then combine that into one single sample and submit that to the lab, at least 4, and that should give you a decent statistical average of your field. For cured flower, this is one where you're going to see a lot of variability, and so I would definitely, once you have something cured and bucked in a bin, 4 to 5 random flowers combined into one sample would be the same. And you don't have to combine them. You could throw them all into a bag and submit them to the lab and let the lab do that. When you have something that's sold more like volume or by volume, the assumption there is that you've done a lot more of the mixing and homogenizing. So it's less of an issue that we've seen with things like oils and creams where you've already heated them and mixed them. Products sold per unit, particularly gummy bears, they don't all weigh the same. So, submit several, and that gives the lab the opportunity to either combine them all and pulverize them into a mixed powder, or to just, mix or to melt 3 or 4 gummies into a single sample. And that way you get a more representative average, particularly when you're talking about how many milligrams of CBD per gummy. They do not all weigh the same, for sure. These amounts are just for cannabinoids testing. So of course you're going to do full panel and a whole bunch of other services. Check with the lab to see how much larger of a sample they want. It would certainly be larger than this. But one thing I want to highlight, when possible, send extra. I know that these are precious materials. I completely understand that. If it's not something that is extremely precious, and I understand that there are some products that are very valuable per gram, and certainly labs should be able to deal with that. But we do things other than just take 1 gram and test it, right? We have quality controls. We'll spike, say you're doing a pesticides test, we will take some of the sample and actually take pesticides and spike them, and then extract that to make sure that we can recover them. And it's just part of the quality controls. Also, sometimes we mess something up and it doesn't work out and we need to repeat it. So if it's possible, send extra. And that really applies to any kind of sample that you submit. In terms of how to submit, we've found that paper bags are the best. If you put something in a plastic bag, and particularly for wet flower, I haven't seen too much difference in the actual results in terms of cannabinoid levels, but there's a lot of moisture buildup in the plastic. If you're using the Postal Service right now at its current rate, it will definitely be moldy by the time it gets here, and it's just not. It's not great to work with. But also the interesting thing about the paper bags is it gives us a head start on drying your sample if it's wet. I get a lot of questions about other sample types and types of containers. We've gotten pretty much any container you can think of, and they all work just fine. So anything that is secure in terms that it won't leak, particularly if you're sending multiple samples, you don't want them mixing with each other. And then this one, clearly labeling each sample is very helpful. Sometimes it's very hard on our end to tell. And so, please definitely clearly label them, and also label them as something that means something to you. Even if you're submitting, you're tracking your THC in your crop, and it's cherry wine. If you call it cherry wine every time, at the end of the season, you're going to have 4 or 6 COAs that all say cherry wine. So maybe say cherry wine week 2, cherry wine week 4, something like that. And do your best to complete all paperwork. We all have something different that we're asking for right now. Chain of custodies, client forms, these kind of things. A lot of them are, mine certainly included, they're a little bit tedious, but there are reasons behind that helps us, track, keep everything straight. And also, being an ISO accredited lab, there are a lot of the paperwork, to be honest with you, is part of the legal backing that is going to support your COAs in the unfortunate event if something had to go to court. You really want all that paperwork tight. And so when it comes to shipping, if you can, hand deliver. If it's a 2-hour drive, I would even consider doing that anyway. Right now in particular, we're seeing a lot of problems. The only service that we have not had any problems with so far has been overnighting through the Postal Service. UPS has actually been pretty good, but they have confiscated some things. FedEx 100% of the time will confiscate anything that smells like cannabis. USPS, just the regular option is really running behind right now. Recently, someone shipped something from around 90 miles away from me on a Wednesday, and I got it the following Monday. And so there's a huge delay. And again, being a fresh sample sitting in the truck for, what's that, 5 days, it's not ideal. Next choice, hand deliver. Like I said, that's definitely the best right now. Postal Service, I'm sure you've heard on news and things, there are things going on there. That does seem to be leaking into hemp delivery to your testing lab as well. So what to expect? I'm just going to try to be realistic with you. There's a lot of sample to sample variability, and it's different depending on what kind of sample. So when I say flower to flower up to 50%, this is why when I talk about cured flower and taking 4 to 5 random samples, this is why, because we tested an entire plant, and the cannabinoid levels at the bottom were 50% of what they were at the top. So you're just basically trying your luck if you submit one single flower. So the hope is, if you submit 4 or 5 flowers and we grind them together, maybe you get a representative sample. That goes down when it comes to products that you have done some sort of mixing on. So if you have a bunch of ground biomass, oil-based products, I would expect variability less than 10%. Individual products, I'm talking more like gummy bears, it just kind of depends on your process and how consistent that is, how consistent you've mixed the gummy material, how consistent you're making the same size gummies and that kind of thing. Lab to lab variability right now, I would say 20 to 25% would be reasonable given the current state of the industry. It's a young industry. It's nothing particularly wrong with it or wrong with the labs and what we're trying to do or why we're doing everything different. It's just an immature industry right now. We don't have standardized methods. And so every single lab is basically doing at least some part of their testing procedure different. That will over time become standardized, and the lab-to-lab variability will decrease, but it will not go away. Right now, different extraction methods, I would say, is probably one of the biggest things leading to variability. And consider when you send like a beeswax-based product, we have to get everything that you added in, we have to get it back out. And different methods have a lot of different efficiencies at doing that. And also there are different ways of calibrating. And so not to get in too far into those weeds, but there are ways to calibrate just with known standards in terms of where it's just like a CBD solution. And then there are ways to calibrate, say like with CBD in a beeswax, and different labs will do different things. And so that could also lead to some of that variability. So hopefully that's helpful. And if you have any questions, my contact info is on here. Happy to expand on any of these if you're interested or help out in any other way I can. But thank you.